So this is question 2 of the FRQs for the 2023 exam. This is another type of like graphing analytical question. They're just giving you a chart here. So let's go through and read the description. It says elevated levels of CO2 increase the rate of photosynthesis and growth in plants. Scientists studying the mechanism involved in these increases examined a variety of species and found that when plants are exposed to elevated levels of CO2, there is an increase in the number of chloroplasts per cell. To investigate whether the elevated levels of CO2 have a similar effect on the number of mitochondria in plant cells, the scientists then selected 6 of these species to quantify the number of mitochondria per cell when the plants were exposed to both normal and elevated levels of CO2 in table 1. Okay, so for this chart, just to break it down, you have 6 different types of species of plants. So you basically have 6 different types of plants that you're testing, and you're exposing them to normal CO2 and then elevated CO2. So you're putting them in the regular environment, and then what happens if I expose them to extra CO2? And what you're measuring in this case is the number of mitochondria, because the number of mitochondria should relate to, how much energy is being made, right? The more chloroplasts you have, the more photosynthesis, the more mitochondria, the more energy you're going to be able to make. Okay, so they're basically seeing whether elevating CO2 is going to increase metabolic activity through measuring how many mitochondria there are. Okay, so let's start off with A, which is pretty straightforward. It asks you to describe the role of the inner mitochondrial membrane in cellular respiration. So this doesn't really have much to do with the question. This is a theory question that you guys should all know. The inner mitochondrial membrane. So if we remember, I'll draw a diagram. You don't have to draw a diagram for the answer, but just for the sake of this, you have your mitochondria, which is a double membrane, right? So this is the inner membrane, and then we're going to do an outer membrane here. They're asking you the purpose of this inner membrane right here. Now, there's a couple different things that you could say, but the main thing that this is going to do is this is where our ETC is located, so our electron transport chain. So your electron transport chain, actually, all these proteins are integral proteins within the inner mitochondrial membrane. So they align all this. And we know that they're going to pump hydrogens outwards here, and what's going to happen is we're going to create a gradient because of that. So the hydrogens are going to be pumped from the inside to the inner mitochondrial space to build up this H+ gradient, and that proton gradient is going to drive ATP synthesis through ATPase. Okay, so for this one, there are a couple different ways you can answer it, but that's the main gist of it. You can say that this is where the ETC is located, so I'll write all the possible options they accepted. ETC located. You could have also said this is where oxidative phosphorylation happens. You can say it allows the building of a gradient, because if you didn't have that inner mitochondrial space, you're not going to be able to produce your gradient. Okay, so because you have 2 membranes, that space in the middle can be used to make a gradient. So it aids in the ability to make a gradient. And then the other option that you could have said is a little bit more vague, but they accepted it as well, is the fact that you would be able to have so the point of the inner mitochondrial membrane is to separate what's happening in the matrix from what's happening in the inner mitochondrial space. So that's another option that you could have done. Let me just expand this. Okay. So you also could have said it's there to separate matrix and inner membrane reactions, which is pretty much establishing the proton gradient. This was only 1 mark, so you only had to say one of these. And again, this is all very basic about metabolism that you learn. Okay, so the next one looks like we're going to be constructing a graph. So it says, using the template in the space provided for your response, construct an appropriate labeled graph that represents the data in Table 1. Determine which species shows a difference in the number of mitochondria between normal and elevated levels of CO2. Okay, so for this one, we're going to construct a graph. They gave you probably a it looks like they give you a space provided. They're not going to give you anything other than basically a sheet of graph paper. So you have to be able to use what they give you to make your graph. They're not giving you the x-axis. They're not going to give you the y-axis. They're not going to tell you whether it's a dot plot, a bar graph, or anything like that. They simply give you graph paper. Okay, so just because I don't have, the actual graph paper here, we're not missing out on anything. So we're making a labeled graph. So the first thing that I want to do is we're going to look at our independent and our dependent variables. Okay. So our independent variable is always what's going to be changing, and our dependent variable is going to be what we're measuring. So what we're measuring in this case is the number of mitochondria. What's changing? The species. So we're looking at different species and the number of mitochondria that they have in two separate conditions. So this is where it gets a little bit complicated because you're looking at two separate conditions. So the way that I would do this graph is, let me go like this. So my x-axis is going to be my independent variable, which is species. And then my y-axis is going to be number of mitochondria, so what we are measuring. Mitochondria per 100 picometers squared of cell area. Okay. Picometers squared of cell area. I'm writing this down because you do have to put the units, okay? Obviously the units are given to you in the table, but you can't just put number of mitochondria. That's an improper axis label. You have to put the unit, okay? So in this case, it's number of mitochondria per 100 picometers squared of cell area. If you don't know what that means, that's fine. They give you the measurement in the table. Species does not have a measurement, so you don't have to worry about that. Now, for the way that I want to do this, notice how I didn't put number of mitochondria for our two conditions. So for normal CO2 and for elevated CO2. Now, what many people may think that they want to do is like a line graph or a dot plot. That is not going to be the best way to do this because you're looking at two separate conditions for separate species. So your species are going to line here. I believe there's 6 of them. 2, 3, 4, 5, 6. And you have 2 measurements for each. So what I'm actually going to do in this case is a double bar plot, okay? Where for each species, I'm going to put down what the number of mitochondria are in normal CO2 and then for elevated CO2. Okay, so I'm going to show you what that first one looks like, and then I'm also going to draw our axis. Ooh, I have to do the scale. So another important thing when you're doing this is you have to write the scale on the side. Be mindful of how much space they give you on the graph paper, because obviously you're not going to extend past that, and don't use just a quarter of it. You want to use up as much space as you can. The way that I typically determine my scale is I look at the highest number and I look at the lowest number. So in this case, our highest number seems to be 2.4, and our lowest number is, I think, 0.3. Okay, so in that case, I would either go up by 0.1 or 0.2. So our highest is going to be 2.4. So I'm going to put 0 here. And then we'll do 2.0 here, 1.0 here. It's a little crowded. And then you can draw the ticks in between. So I might draw 10 ticks. Yeah. 1, 2, 3, 4, 5, 6, 7, 8, 9, and then 10. So this would be like 0.1. You don't have to write all of these. 0.2, 0.3, 0.4, 0.5, 0.6, 0.7, 0.8, 0.9, and then 1. Okay, so now I want 10 ticks in between number 1 and number 2. Okay, I fixed this up a little bit so that you can see the differences. And so I'm gonna add 10 tick marks between 1 and 2. For this, I'm doing this because I don't have graph paper. For this, make sure that there's 10, spaces in between. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, and then 1, 2, 3, 4, 5. We can just go up to 2.5. It's fine. So this would be 2.5. Alrighty, so now we're going to draw our bar plots. So if we start off with species number 1, you've got for normal CO2, 1.0, and for elevated CO2, you have 1.6. Okay, so there are 2 measurements, and like I said, I'm going to do a double bar plot. So this is for species 1, and I'm going to have 2 bars right beside each other. The first one is going to go up to 1, and the second one is going to go up to 1.6. So this one's going to go up to 1. And again, I'm doing this on my iPad, so it's not perfect. And then 1.6 should be 1.2, 0.3, 0.4, 0.5, 0.6. Should be about here. Like that. And so a really important thing that you have to remember for these double bar plots is a legend. Because you have 2 bars per species, you need to be able to identify which one is for elevated CO2 and which one is for normal CO2, right? Because we're comparing this to this. I need to know which one is the elevated CO2 and which one's the normal CO2. So let's just do the elevated CO2, which should be the second one. We'll just color it in. So that's how we're going to separate them. You can do something simple like coloring it in, stripes, whatever it may be, as long as you put a distinct difference between the 2 bars so you can see them separately. You would also have to do a legend because of this. So my legend would then look like this, where I have a blank square like this, which is going to represent normal CO2 environment. You will 100% lose marks if you don't do this, okay? And then I'm going to draw another square and color it in to show that that represents increased CO2. And then the last thing for this is they also want you to add standard error bars. Okay, they gave you standard error here because they want you to use it. So as you can see here, for the normal CO2, you have 1.0 plus minus 0.1. So your standard error bar in this case is 0.1. They won't, be very meticulous with this, and they're not going to take a ruler. So as long as it's close to a 0.1 error bar, then you should be good. So I'm going to do my error bar in a different color just so we can see. So the error bar for this one was 0.1, meaning it could be 0.1 higher or 0.1 lower. So my error bar, I'm going to go up by 0.1, and then I'm going to go down by 0.1, like that. The second one, it's also going to be 0.1, so I'm going to do the same, up by approximately 0.1, and then down by approximately 0.1. So that's species number 1. Species number 2, we have 0.4 plus minus 0.05. So this one will go up to 0.4, 0.1, 0.2, 0.3, 0.4. And then plus minus 0.05. So that should be a really small error bar. Around like that. And then for increased CO2, it is 0.9 plus minus 0.08. Again, this is a hard number to quantify. They're not going to be super picky. As long as the error bars are there and they're like the relative right size, then you should be fine. So 0.9, which should be around here. So not quite at 1. And then plus minus 0.08, so I'm going to make it a little bit smaller than my 0.1 error bars. Like this. And remembering to color this one in, because this is our increased CO2 one. Number 3, 0.5 plus or minus 0.07, so 0.1, 0.2, 0.3, 4.5 should be around here. Plus minus 0.07. So it should be plus or minus close to a 0.1. Okay. And then with the increased CO2, it is 0.9 plus or minus 0.1. So 0.9 should be around here. I think the other one's 0.9 as well. So they should be at the same spot. Plus or minus 0.1, so you're going to go up to 1.0 and down to 0.8 in this case. And making sure, again, to color that in. Species 4, 0.3 plus or minus 0.03, so 0.3 is right here with a really, really little error bar. And then elevated CO2, 0.6 plus or minus 0.06. So 1, 2, 3, 4, 5, 6, around here. And the error bar is 0.06, so not quite 0.1. And then again, color that in. 5, we have 0.7 plus or minus 0.06. So 0.7 should be 1, 2, 3, 1, 2, 3, 4, 5, 6, 7. So it should be a little bit higher than obviously our 0.6. Error bar. And then increased CO2 is 1.5. So that's a pretty big number. Plus or minus 0.22. So these should be much bigger than your other error bars. They're like double the size. So make sure you show that. Again, like I said, they're not going to be super picky, but if your error bars for 0.22 are the same as your error, like same size as your error bars for 0.1, then that's a problem. Okay. So 1.5 plus minus 0.22. 1, 2, 3, 4, 5 should be around here. Again, this would be easier with graph paper. Just kind of take this lightly. And then I have a really big error bar. So I was at 1.5, 1, 2, 3, 4, 5. I should go up by 2 and then down by 2 for my error bar. Okay? Because it's about 0.22. So I think up by 0.2 and then down by roughly 0.2. Color it in. And then our final one, 1.3 plus minus 0.15. So 1.0, 1, 2, 3, this. Plus minus 1.5, so it should be smaller than this one, but still larger than smaller than 0.22, this error bar, but larger than the 0.1. And then finally, you have 2.4 plus or minus 0.22. So 2.4 should be around here. I'm going to cut into my legend. Yes. Oops. And then your error bar is 0.22. So I actually should have extended this a little bit more. So we'll extend this a little bit more because we need to. 2.6, 2.7. Okay. And then this error bar should be about this big. Okay, so that is your graph for this one. I think that is everything. Oh, so construct an appropriately labeled graph. Determine which species shows a difference in the number of mitochondria between normal and elevated levels of CO2. They're asking you, out of our 6 species, which one are showing a difference? Well, if I take a look here, and I consider my standard error, because that's going to be able to determine it, it looks like all the species are going to be different, right? You can see that none of these error bars overlap, and all of them are going to have a difference between normal CO2 and then increase in CO2. So the answer for this one, you have your graph, and determining which species, I'm just going to say all species show difference. Okay, so that is part B. Took a while, but it's really important that you practice these questions so that you know what graphs to use at which time. Because if you started doing a dot plot or a line graph for this one, you would have quickly realized that it was not going to work, right? Because you have 2 separate conditions and you're only taking one measurement for each. A line graph is going to be good when you're taking multiple measurements over time. In this case, you're only taking one measurement. Okay, so typically bar graphs are going to be good for that. If you have multiple conditions, you just do multiple bars and making sure to have a legend there so that you can determine the difference, obviously, between the bars and what they represent. Okay, so part C is asking you, based on the table, based on the data in table 1, describe the relationship between the level of CO2 and the average number of mitochondria per unit area of a cell. Okay. Relationship between CO2 level and mitochondria. It's ultimately asking you the goal of the question, which is what happens when you increase CO2. So you can use the table easily. We've already modeled it on a graph, which makes it pretty easy to see, given that you did your graph correctly. So I'm going to use this graph here. They're asking you, okay, the relationship between CO2 and number of mitochondria. You can see clearly for every single one of these that when you increase the amount of CO2, you're also going to increase the amount of mitochondria, because this increase in CO2 caused a spike in number of mitochondria. Same thing here. The normal CO2 always has a lower amount of mitochondria compared to the increased CO2, right? Got increased, it got increased, and it got increased, and then it got increased again. There's always a higher amount of mitochondria when you increase CO2. So if I were to describe a relationship like that, let me extend this. It would be a positive relationship, okay? Because increasing CO2 increases number of mitochondria. So you could say positive correlation, or you could just say increase in CO2 causes increase in number of mitochondria. Okay. So it's really just asking you to identify that. You should know the relationship, so what a positive relationship means, or a positive correlation and a negative correlation. In this case, it's positive. They would have accepted either. So if you said positive relationship or you said number of mitochondria is greater with elevated CO2, they would have accepted either. But just in case, remember what a positive and a negative correlation are. Okay, so for the last part here, it looks like a multi-part question. So we're gonna scooch this down and tackle this one. Okay. So D says the leaves of a particular plant species are typically green. But scientists notice a plant in which the leaves have white stripes. They determine that the stripes result from a mutation in mitochondrial DNA that interferes with the development of chloroplasts. Okay, so we've got some plants with white stripes. That's going to be as a result from a mutation in mitochondrial DNA. That's going to be very important, and it's going to interfere with the development of chloroplasts. Scientists cross plants using pollen from the plant with white striped leaves and ovules from a plant with green leaves. Okay, this is also very important. So, pollen from a plant with white striped leaves and ovules from a plant with green leaves. Predict the phenotypes of the leaves of offspring produced from this cross, and then provide reasoning to justify your prediction. Explain why. Okay, so I'll do that after. Okay, so what we basically did here is we took pollen from the white striped, pollen, white stripe, and we crossed it with ovules from the regular green plant, yeah. And they're asking you, what are the offspring going to be? Now, the fact that they have given you no concept of dominant or recessive should give you a little bit of a hint here, in that fact that you don't have to do a Punnett square. It's impossible to do a Punnett square for this one. It's a theoretical question. And so the key to this type of question, or the key to this one, is that the mutation is in mitochondrial DNA. They tell you this in the question. It's due to mitochondrial DNA. Now, might not be easily found in the curriculum, but I like to teach it specifically for this question, just in case something like this comes up. The key thing for this one is mitochondrial DNA is inherited maternally. This is a very, very small detail in the span of the curriculum. So it's something I always reiterate to students just in case this type of inheritance comes up, because I don't think we do mitochondrial inheritance patterns too much, at least for pedigrees and stuff like that. But the key to this question is that mitochondrial DNA is inherited maternally. For plants, for animals, everything. So what that means is the mother is going to pass on those genes, okay? Let me move this. Okay, so when a mom has a condition and she has offspring, she is passing all of her mitochondrial DNA to her offspring. If your mother has a mitochondrial disease, it's very, very likely you will also have that mitochondrial disease. If your dad has a mitochondrial disease, you will not be affected by it. So you are technically more related to your mom than your dad, because they split the regular chromosomes, 50-50, and you get all of your mother's mitochondrial DNA. So given the fact that this mutation is inherited mitochondrial DNA, it's going to go based off what the mother is. So for this one, you have pollen versus ovules. This is a difficult question, I will say, if you're a little bit lost. I was very surprised by this question, because they also don't really teach too much about like plant reproduction, but pollen is the male, and ovules, you can kind of assume, because ovules kind of sound like ovaries, are the female. Okay, so the green plant is the mother in this case. Therefore, all of the offspring will be green. Okay, the dad in this case does not give away mutations because his mitochondrial DNA is non-inherited. Whatever the mom's phenotype is, that will be the children's phenotype. In this case, the mom is the green plant. Oops. So all of the offspring will be green. So it says, predict the phenotype of the leaves of offspring produced from this cross and provide reasoning to justify your prediction. So mitochondrial DNA is inherited maternally. So you can say, therefore, since mom is green, all offspring will be green. Okay, I believe there is another little part to this question. It says, explain why plants with the same genotype are able to differ in the structure and or number of certain organelles in response to changes in atmospheric levels of CO2. Okay, so this is again more of like a theoretical question. And it's asking you, okay, so this is related to the specialization of cells and what we talk about in Unit 6. So why do, for example, I'll give you an example that's not plants because we don't usually spend too much time on plants. All of our somatic cells in our body have the exact same DNA, right? We came from one cell that multiplied. All of our somatic cells have identical genetic information. So why do we have brain cells versus heart cells versus liver cells? My liver cells have, different amounts of organelles than my heart cells, right? The liver cell, like for example, I'm just giving you an example. The liver cell has a lot more smooth ER because the smooth ER is really good for detoxifying, which is the job of the liver, right? So there's an example like that. Ones that are really going to be important so your muscle cells are going to be really good at metabolism because that's where you're producing your sugar. So the way that's going to be controlled is through gene expression. So all of your cells have the exact same genotype, same genetic information, but the reason some have more organelles than others, not different organelles, they'll all have the same organelles, they'll have different amounts, so more mitochondria versus less mitochondria, is because of gene expression. So for this one, I would say that CO2 concentration affects gene expression. Okay, and as a result, you're going to have different amounts of mitochondria for each. So I think they would have accepted changes in CO2 levels affect the expression of certain genes. Okay, and therefore that's going to affect the amount of organelles you have. Alrighty, so let me just see how much this one was worth. This one was also worth 9 points. Okay, so these were your two big questions. Usually you have your two really long ones at the beginning, and then 3, 4, 5, and 6 are usually shorter, around 4 or 5 marks each. So this would be 1 mark. How much is the graph worth? I'm just doing this so that you guys can mark yourselves. The first one is obviously 1 point. This one is going to be 4 points. The graph is worth a lot of marks. You usually get marks for the axis, the scale, proper data points, and then probably the legend is the 4th one. And then this one is 1 point, and then this one is 3 points. Okay, so yeah, that all adds up to out of 9. So that is question number 2.